All right, we're going to go through the process of mitosis here. It's worth noting a couple things. One of them is that mitosis is only one of the parts of the cell's cycle coming after interphase, which has growth 1, synthesis of DNA, and then growth 2. It can also be compared with binary fission, which is much, much simpler, and in prokaryotes, where they basically replicate their DNA plasmid, they elongate just a little bit, and they pull one plasmid off to each side, kind of just by chance, and there will probably be several of them on both sides, so that when they build a new cell wall or split, there's DNA on both sides. It's also good to review some of the names of the structures inside of the cell here. Of course, we've got the cell membrane. Replicated DNA is connected by a centromere, and the two identical copies of that DNA can be called sister chromatids. So connected with the centromere, not to be confused with the centrioles over here, which are sending out spindle microtubules, and then these objects together, the spindle microtubules and the centriole, can be collectively called a spindle. So in interphase, the cell is going about its business, producing organelles and proteins. The DNA may be copied. The DNA at this point is stringy, as is noted in this diagram, and usually you would not need to draw a cell that is not going through some sort of division process, and therefore not need to draw stringy DNA. Prophase is where easily the most things happen. You'll notice that the nuclear membrane is actually disintegrating around the DNA, which has now condensed into chromatin fibers and then also into chromosomes. So the chromosomes are now visible. The centrioles have replicated, and they are moving off to opposite poles of the cell, and they're sending out microtubules, and that is about it for prophase. In metaphase, those spindle microtubules have grabbed on to the centromere, and specifically this little region of it called the kinetochore. And they have lined these chromosomes up into the middle. Notice that they are still called chromosomes at this point, made out of two sister chromatids. They should be lined up in the equator of the cell, and that can be referred to as the metaphase plate. In anaphase, the centromeres divide and the sister chromatids are pulled apart. You can actually now call each sister chromatid a chromosome of its own. It's almost like it graduated to the term chromosome. So this movement is going to bring these chromosomes off to their respective poles, and it will free up the middle so that when cytokinesis happens, hopefully no chromosomes get caught in the middle. In telophase, you basically undo everything that you did in prophase. The nuclear membranes start to come back, the chromosomes will start to uncoil into stringy DNA, the spindle begins to disappear, and the cell elongates a little bit, and then other parts of the cytoskeleton, these tubes that are inside the cell and help to change its shape and maybe move the cell a little bit, that pinches in the membrane right here, and as soon as the membrane has been completely pinched off so that the cytoplasm is divided between the two cells, that's when you can officially say that it is split in two and that mitosis has completed. So here it is sort of in the whole scheme of things. Note that interphase is not actually a part of mitosis, and there we have it. These can be drawn with relatively simple drawings. Keep in mind how many chromosomes you ought to have in the cell if it's specified, but you don't need to be too artistic if you were to diagram this process.